You all for the kind attention and the chair for the word. I'm going to present you today this work regarding actually the exposure of bacterial and fungal communities to heavy metals into three different environments: the soil, the sediments, and the rhizosphere. This work, first, let me say it deals with biodiversity, and now biodiversity is becoming day by day more important. As we can see here, it is being introduced actually into constitution, so into politics. And biodiversity, we have many different metrics to measure it. We have alpha diversity, we have beta diversity, and gamma diversity. This work deals with alpha diversity. And the alpha diversity into environment, its variations, particularly for macroorganisms, are usually associated with the environmental perturbation. And low levels of alpha diversity are generally associated with pollution, with the environmental contamination. One particular contaminant that we are focusing on today is heavy metals. This class of contaminants is a special class because they cycle into the environment due to human activity, and they cycle up to the point they reach compartments where they sink in, they accumulate, and they exert toxic effects like soils and sediments. Soils and sediments harbor huge amount of biodiversity. We have thousands of different taxa belonging to bacteria, fungi, or even soil fauna. But while the effect on soil fauna is nowadays clear, so the effect of heavy metals increasing the pollution with heavy metals, we have a decrease in the alpha diversity of arthropods or earthworms or even bees, fossorial bees. So those bees nesting into the soil. The thing for microorganisms is not so clear. We do not have particular clues on what is doing actually microbial alpha diversity when the doses of heavy metal, when the content of heavy metal is increasing into the environment. And this is still an open question. So to respond actually, to answer to this question, we performed a meta-analysis. A meta-analysis, just I'm gonna be brief, is just a method that research has to synthesize research on a precise topic, and so giving one single interpretation of the data generated by huge amount of literature. And in our meta-analysis, we considered the pollution of soil, sediments, and rhizosphere, as the title hinted, in when considering actually copper, cadmium as heavy metals. Why copper and cadmium? Well, just because they are very much used in agriculture, for example, in industry, so technology and industry, or for example, human driven activities like smoking, waste management. And we discarded all those studies that were not focusing on the screening of bacterial and fungal communities through next generation sequencing approaches. So those studies, we set that the time lap in between 2013 and 2022. Of course, the studies must taken into consideration the alpha diversity as a metric to estimate the response of the microbial communities. This produced a strict selection starting from 11,000, almost 11,000 studies screened. We finished with 32 studies included. Why? Because we discarded daily techniques, less sensitive techniques. We discarded also studies because of methodological issues, or, for example, because of the fact they were not describing so well the doses that were added to those environments. And also we discarded those environments those studies that were focusing on already polluted environments, because we were just synthesizing studies performing those perturbation experiments. And this perturbation was tracked down, computing an index, tracking down what was the response of alpha diversity on the treated samples in comparison to the control sample. And let's jump into results. We have the majority of the studies conducted in the People Republic of China, and the sample size is varying from 2 to 8 replicates. But most importantly, we have the metal addition that is spanning from 0.3 milligram kilos to 5,000 milligram kilos. I want to stress this point because 5,000 milligrams of an heavy metal, so like a spoon of heavy metal added into 1 kilo of soil is actually an extreme amount of heavy metal, just because those doses, those contents actually of heavy metals can be found in mining ores. So in those sites where the heavy metal is such highly concentrated that we can actually go there and take it out of the soil. We then performed a best model selection considering all the parameters that we extracted from the works. So like the form of addition of this heavy metal, if the matrix was fertilized or not, if it was amended or not, the environmental parameters like pH and organic matter that would have modulated the availability of those heavy metals added. And surprisingly, none of these parameters were actually influencing the main response. That was also the best actually model describing what was called the effect size, so the variation of the alpha diversity from the control samples. And here we come to the main result. We have that the relation actually between the metal dose and the alpha diversity change from controls is negative. This means that increasing the metal dose, the alpha diversity decreases in comparison to the control samples. But this must first be exceeding 17.5 milligram kilos as minimal dose. But also, this is a moderate decrease if considering the extreme doses where soils were subjected. We have 500, 1000 milligram kilos that are mimicking polluted soils like agricultural soils, and they cause actually 10-11% decrease in alpha diversity. But also peaking to 3000, 5000 milligram kilos, we have only 13-14% of decrease. And this decrease, you see on the bottom right, it was not as expected. It was not dependent on the alpha diversity metric considered. They were all concordant in describing this decrease. Also, the experimental condition as discarded, as you have seen before, discarded by the model selection, here we have a clue on that. We have no influence of those covariates, let's say, on the main response, on the metal the metal dose dependent response. We have no influence of the pH, of the organic matter, of the form added, or the presence of microbial communities in a specific environment, whether it was soil, sediment, or rhizosphere. So let's jump into conclusion. That still open question, now it's not so open, because we found out that there actually is a negative relation between heavy metals and microbial alpha diversity, but, and this is a but, this response is not so big if considering the high extreme heavy metal doses we were considering. And this is another puzzle piece highlighting how the microbial world actually differs from the macroorganism world. And those rules, those ecological rules that we apply to the macroorganism world are not so easily extrapolated, cannot be so easily extrapolated and fit into the microbial world because they behave quite differently. Also, this is an hint to find new metrics that can describe what really happens to microbial communities, like here in heavy metal pollution, in heavy metal perturbation conditions. So yeah, now I finished. I want to thank you for your attention, and also the lab in which I'm a PhD student. And yeah, so if you have questions, I'm here.